Thank you. So hello, everyone. My name is Brianna, and I will talk about Laboulbeniales, which is a group of fungi and ladybirds. And it's perfect. Okay, so first, a little bit about myself. I am from the United States. I grew up in Florida. I grew up in a small beach city down here, where it's always summer and completely different from the weather here. And then for my bachelor's, I moved up here to Orlando, Florida, which is actually where Disney World is located, if anyone knows anything about that. And after my bachelor's, I actually flew all the way to Ghent, Belgium. I was funded by the Belgian American Educational Foundation. So I went currently here to get my master's at Ghent University, and I'm part of the research group mycology, which is led by Professor Dr. Enrica Verweken and my own mentor, Dr. Danny Hellwaters. So my research interests are in entomopathogenic fungi, so fungi that parasitize insects. I'm interested in a wide variety of ecological questions, such as how do abiotic or biotic factors affect this relationship, how do other organisms affect bees, parasites, etc. So my current research project and what my master's is on is this group of fungi called Laboulbeniales. So Laboulbeniales is an order of fungi in the phylum Ascomycota, and they are obligate ectoparasites of arthropods. So they complete their entire life cycles on the integument of arthropods, and they spread primarily through touch. So sometimes you get these like really beautifully infected organisms, such as this bat fly right here, super infected. These are what these fungi look like when they're off their hosts. So they attach to their hosts using this cell right here, this is called the foot cell, and then they grow into these elongated structures that we call a phallus. And here you can kind of see that this structure right here we call the perithecium has all these sticky spores in it. So when these spores, when the perithecium comes into contact with an insect that its host, then the spore will be released and it will stick on to the integument of the insect, and then it'll penetrate it sometimes and grow all on the outside. Some of them do have these penetrating structures, like this one has a haustorium, so it's a root-like structure that can actually go into the integument, and some of them don't actually have that, like this. And as you can see, they come in all sorts of shapes and sizes. Like this one is super small. This is the one that we like to show a lot. It's called Laryngomyces plesiosaurus and infects bat flies, and it looks like a plesiosaurus. So this one I'd like to draw our attention to. This is called Hesperomyces resins, and it's the one that I currently study. So Hesperomyces resins infects ladybirds. Here's a nice picture of them on the elytra of one of the ladybirds, and then here's a drawing of the structures. And as you may have guessed from the title, this is actually a species complex. So each Hesperomyces species infects its own genre of ladybirds. So the Adalia all have their own Hesperomyces species, while the other genera also have their own. The one I'm specifically looking at is down here, that infects Harmonia axaridis. We call this Hesperomyces harmoniae. So I'll change gears a little bit and talk more about Harmonia axaridis before I talk about Hesperomyces. So Harmonia axaridis, also called the harlequin ladybird, is this pretty ladybird right here that comes in a variety of different colors. These are all the same species. Now, these are beautiful, but they're also kind of monsters because they're really highly invasive. So these are all native to Southeast Asia here. And in the 1900s, they were brought to the US and then Europe as a biocontrol for aphids. They're really good at eating aphids and other plant pests and keeping them away. But unfortunately, an invasive population established itself in the US, and then from there it would spread throughout the entire world. through anthropogenic means to become a globally invasive species. Now, in its non-native habitat, it's been known to outcompete and even eat native ladybirds and cause dramatic shifts in the structure of the community populations, especially in the UK. There's been a lot of studies about how they're outcompeting native Adalia and other ladybirds. So with this in mind, researchers have been looking for a way to control invasive populations, which brings us back to Hesperomyces famoniae. So here is a map from iNaturalist that I downloaded a few days ago. And this is the distribution of Hesperomyces famoniae as shown by the observations. And as you can probably see, this isn't the full distribution, this is just on my iNaturalist, but it really follows closely the distribution of Harmonia here. So Hesperomyces famoniae really follows and infects Harmonia axaritis where it is in its non-native ranges and sometimes in its native range. And then here, this was a study done in 2022 on the prevalence of Hesperomyces famoniae on Harmonia axaritis in Europe. And as you can see from these numbers, this is the prevalence in percentage. Some of them can be as high as 46%. So a significant proportion of the population is actually infected with this fungus. So what do these things actually look like on the ladybird? Well, we see, before I explained that they spread through physical touching. So sometimes we get really sex-specific positions. So here, this is a female ladybird. We mostly find it infected on its elytra, while here, a male ladybird is mostly find it infected on its abdomen, and that's because they mate like this. But ladybirds also aggregate together during overwintering. So they form these structures where they all clump together, and this can further spread the fungus. So we can get really highly infected ones like this. And then I'm just going to go over a little bit about the effects of Hesperomyces famoniae, what we know so far, and its potential for biocontrol. So we know that it can cause increased mortality in ladybirds, especially male ladybirds. But then during winter, it can cause even more mortality, so they'll die more during overwintering. When we looked at their immune responses, we actually didn't find any significant changes in their immune responses, so the fungus isn't triggering their immune system. But we did find a large change between their pre-overwintering body mass and their post-overwintering body mass, which leads us to believe that maybe they're dying because of energy exhaustion. And then the next few studies, we did a lot of dual infections. So we infected ladybirds with Hesperomyces famoniae and other pathogens. So when we used other enteral pathogens like the Bayer Bassiana, we didn't actually find any increase in mortality in Harmonia. And then when we use a bacterium, Spiroplasma, which is known to infect and kill ladybirds, we actually found a decrease in the number of eggs that they hatched, and then also a decrease in the number of larvae. And then finally, when we infected them with a parasitic mite and Hesperomyces, we actually got increased mortality, but mostly in male ladybirds. So this is what we know so far about what Hesperomyces famoniae does to Harmonia. And there's still a ton of questions left to answer, but this is a few that I found the most interesting. So obviously, first is biocontrol, if we can actually use this to help control invasive populations, if we can combine it with other things to maybe increase its mortality to a rate where we can actually use it for anything useful. Second question is really important. So what does this fungus feed on? We actually don't know how this fungus attains its nutrients. The prevailing hypothesis is that it might take up lipids from the elytra, but we're still not completely sure how it does that. And then the third question is what abiotic factors influence infection? How does the effects of the environment cause infection in the ladybirds? And this third question is actually the one that's the topic of my master's thesis. So what I'm doing here I am basically infecting ladybirds in the lab. I'm placing them in incubators with different temperatures and humidities, and then I'm monitoring every day to look at the growth of the fungus. So how exactly do we infect them? Well, we actually do have this really super cool technique that we use. This is a hot dog roller. And we put 4 to 5 different ladybirds, or 4 to 5 ladybirds in these little tubes here, along with one really infected one. And then we kind of just spin them on that for an hour, put the heat off, obviously, or we kill them. But this helps ensure that all ladybirds are in contact with the phalli so we can spread the spores and they'll be infected when we use them. And then here's some pictures of my experiments. This is what they look like in the incubators. We put them in these open petri dishes so they can actually be able to follow effects of different humidities. This is what some of them look like underneath the microscope. It's really difficult to see for most of them. So right here, it looks really dirty, but it's actually pretty infected, and these are immature fungi right now. This one is also immature and very small, so it's hard to see. This one is these ones are easier to see, but you really see a full cluster of 3 of them that are really balloon shaped. So I will end this talk talking about reasons to study this group. So this group is small, there's not a lot of people studying it, so you kind of have to justify to people why you're looking at something like this. So the first point is parasites themselves are understudied. I think anyone who is in parasitology can tell you that they're often overlooked despite being very important for the ecosystems they're part of, the communities. The second point is there's a lot of hidden diversity that we don't know about this group. They like to infect really small insects, and they're also very small themselves, so they're very hard to notice. So there's probably a lot of species that we don't not even sure can be infected yet, or are infected that we don't know about. And then the last one, we can also use Laboulbeniales for other practical uses. So I think one of my favorite uses is that since they infect where they touch, then you're going to find infections where organisms touch each other the most. So on roaches, we usually see it on the antennae because they like to touch their antennae together to recognize each other. But then for stuff like the ladybirds, sometimes where you see them completely covered, you can tell that these organisms clump together. So I think that's a cool practical use. If you see a lot of organisms that are infected consistently in one spot, then you can infer that maybe that's where they usually spend the most amount of time touching each other. And that is the end of my presentation. It was a little short, but thank you for coming to my talk. And I know Olda isn't here, but I am grateful that he invited me here just for the summer. And I've had a lot of fun in Czech. It's a really nice country. I really liked your kielbasa and my crusader. And that is all. Thank you.